All right, everyone, we're going to get started. Thank you so much for being here. There's a couple extra chairs if you need them. We can move them around, get everybody where you can see. Thank you so much for being flexible with me, for folks in the room. We are in a different room than we normally are, and you all know that. For folks online, you might notice the camera angle is a little different. We've had some technical issues, but we are all here together, and I think it's going to be wonderful, so thank you for being here. My name is Cinnamon Moffett, and I'll be your host for today. This is a hybrid event, so folks online and folks in the room, we will manage your questions together, just like we do in the seminar room. If you have questions at the end, just raise your hand, and we'll bring a mic around to you. And for folks online, if you have any questions, just put those into the chat box, and Roseanne, our volunteer, who's still getting set up, thanks Roseanne, we'll read those questions out and we'll get everything answered that way. A couple of announcements that I wanted to make. Next week is a holiday, so we will not have seminar. I'm sure you're so disappointed to not be with us then. But the following week, somebody you all know and love will be giving our talk. So our iLab manager, Drummond Wingrove, is going to be giving the talk next week. He gave me a title and a short abstract, but I left it in the other room after I pressured him. So what is our title?
It is Get to Know Your iLab, from ideation to creation, we can help.
So I hope you all come back after the holiday and join us for that one, so we can have Drummond have his full audience with everybody. But Drummond's also the individual who invited today's speaker. So I'm going to invite Drummond up, and he's going to introduce today's speaker. And again, thank you all for being here and being a little bit flexible.
Okay, I didn't get any bio notes, so I can't just read off of something. So I'll just make some things up. Scott Loranger got his PhD from the University of New Hampshire, and he's actually one of the ones that I would blame for corrupting me into ocean engineering and marine science. So put your blame towards him. From there, he then went to work at Hui.
I was at Norwegian Geotechnical Intel.
Yeah. I'll make things up and then he'll correct me. He really did his job for teaching us what the word acoustician means, and I think that has led through a number of different careers. Going to Hui, I think, is what I remember, and past that, that's on you.
Very cool.
Okay, we're gonna leave it there.
Oh, does that work?
Yep.
Okay, cool. Thank you, John. All right, so like John said, I'm Scott Loranger. I'm an acoustician and application scientist with Kongsberg Discovery, which is a company that manufactures mostly underwater acoustics instrumentation. Prior to that, I was a postdoc at Hui, and before that, a postdoc at the Norwegian Geotechnical Institute. And Kongsberg is a Norwegian company, and that's sort of where a lot of the overlap came. I've used these instruments for the majority of my research, starting in grad school. And so we'll start with the very basics. If I can make the slides advance. What is sonar? And I genuinely am asking. There's participation grade for this class. So who knows what is sonar? Shout it out. Sound.
Navigation and ranging.
Oh, nailed it. Who said that? Nice. Sound, navigation, and ranging. It's actually an acronym for, sound, navigation, and ranging. There's a number of different kinds of sonar. So we kind of step through the different versions of how we use sound in the ocean. There's active sonar, which is what we'll talk about today. So you generate a signal, and then you listen for it to come back. It goes out, it bounces off something. It could be a plankton, it could be the seafloor, it could be a submarine, it could be a bubble. Whatever it bounces off of, you listen as it comes back. Passive sonar is usually what you do with a hydrophone. That's where you listen. So it's a lot of like whale localization and ship traffic. Geological events are typically measured by passive systems. So we're going to focus on active. So what is the main application of sonar? When you think of a sonar, what do you think of it doing? The active version, what is it measuring? Distance, right? It's typically what you use for depth, right? So originally, we would use a lead line to measure the depth of the ocean. That's where you just take a weight on a line, throw it over, and measure how long the line was when it hit the bottom. Now, an interesting fact is when you get to the really deep parts of the ocean, things like the Challenger Deep, which we're going to talk about a little later, when you use a lead line, you have to measure the depth on the recoil of the line. So when you're pulling it back in, Does anyone know why you might have to do that? You can't measure as it's going out. You have to measure as you pull it back in.
Because the line stretches.
Because the line stretches. Exactly. Well, you do a lot of physics problems like waves on a string or a lot of your basic learning physics. You assume that lines have no mass, and that's not true. So if you put out a wire line all the way 10,983 meters deep in the ocean, that's a lot of line. That line has mass, it stretches itself, and you have to measure it as it comes back on from that stretching. So we've moved past lead line a lot. You can also measure the depth with a single beam. That's not really in vogue for measuring the bottom of the seafloor. Typically, you'd use a multi-beam, which is multiple beams. It can measure a much wider swath, a bigger part of the ocean in a single measurement. We're going to actually focus on single beams, because this talk we're interested in what's in the water column. So we're not really looking at just ranging, just figuring out the depth. We're going to talk about stuff in the water column. So if you think of a sonar and something in the water column, what do you typically think you might be looking at? Fish. Fish finders, right? Fish finders also a sonar. In general, there's two different kinds of fish finders. For the most part, this is an overgeneralization, but in general, there's two different kinds. There's the single beam, which is what I'll talk about. And then you see on the side here, there's the omnidirectional sonar. So this generates a wave all the way around the vessel. And this is how you would hunt for fish. It's where you would look to go find a fish. This is more of how you would interrogate the fish that you found. It's one way to think about it. Omnidirectional sonars look like this. This is your Hunt for Red October, your every movie ever. This is what they show you for a sonar. It goes around in a circle, it makes a blippy noise, and you see things as it spins around, right? That's an omnidirectional sonar. Typically not something we use for quantitative acoustics, like I'm going to talk about. For quantitative acoustics, we use split beam echo sounders, which is a version of a single beam. I'll talk about that in a second. And this is what your data looks like. This is your, we call it an echogram. So you have the transmit pulse up here, the bottom of the ocean down here, a whole bunch of fish floating around. I think this is a calibration sphere, which is what we use to calibrate the system and do quantitative measurements. You can measure as a function of frequency. So this is the frequency response of what you're looking at. And this is that split beam technology, which tells you where in the beam something is, which actually ends up being really important. So you take your transducer face and you divide it into 3 or 4 sections, and then you measure the phase of the sound that comes back. So remember, back to physics, the wave has 2 properties essentially. There's the amplitude of the wave and the phase. So where in the wave you are. Are you at a peak? Are you at a trough? Are you in the middle? That's the phase part. So you measure the phase that arrives on the transducer, and that tells you where something is in the acoustic beam. That ends up being really important. So with a single beam, you don't know. You don't know if this fish is over here, if it's straight down, if it's off to the side. And you can kind of imagine that if you're sending sound straight down onto this fish, you get a lot of sound that bounces straight back to the echo sounder. Now, if the fish is off to the side, a lot of that sound is actually going to bounce away, right? Just think about the physics and the angle. That fish is going to appear lower amplitude because it's over here, not because the fish has changed, just because it's changed its position. So less sound comes back to the echo sounder. So if you want to do quantitative work, you have to account for that. You have to account for the fact that the target is off to the side. So the split beam, you know where it is. And now you can calibrate for that. You hang a target with a known backscattering strength. You know how much sound should come back. You move it around, and you correct for wherever it is in the beam. Now you can do quantitative acoustics, which is what the rest of this talk is going to talk about. So breaking this down into sort of the 2 main components of underwater acoustics and acoustical oceanography. So we'll start with underwater acoustics. That's really more of the physics and engineering side. That's solving the wave equation, a lot more of the sort of nuts and bolts side. And then acoustical oceanography, which is actually what I do more of, is the application. It's taking that acoustics, taking what's learned from underwater acoustics, and going out and measuring things in the ocean, figuring out what's out there using acoustics. So next question, why would we use broadband? So that's my interest, is in using broadband acoustics. And to answer that, what is this? What is this a picture of? Cereal. So I heard Rice Krispies. That's a guess though, right? You're not sure. Just point that out. What you see here is a black and white image of breakfast. Right? It looks like cereal. It could be oatmeal. You can convince yourself. Right? But it's probably You're all too close to the screen is the problem. It could be, it's some kind of cereal, right? I'll tell you, I didn't hear what it actually is yet. Oh boy, Cocoa Pebbles, we got Fruity Pebbles. All right, so this is the equivalent of what we call CW sound, where you use a single frequency. You get data, you get information, but not quite enough to get the full picture. You know, we know it's breakfast, we're pretty sure it's cereal, but we don't know exactly what it is. When you introduce broadband, you get more information. And Fruity Pebbles, right? No doubt about it. When you introduce this broader information, the broadband signal, now you know what you're looking at. There's a number of other benefits. There we go. So when you use a broadband signal, and I'll kind of define exactly what that means in a minute. Basically, it's using a lot more frequencies of sound. You get improved signal to noise ratio. So what we do is something called matched filtering in broadband acoustics, where you take your transmitted signal. So in a normal single beam, a single wave, continuous wave, one frequency sound is one note, right? That's the sound you generate. If you want to do broadband, you got to sweep through frequencies. That's the, see if I can do it. Right? You just sweep through a frequency and you cover all those frequencies and you get more information. Now, if you take that unique signal you generated that spans all those frequencies, that chirp signal, and you correlate what you've recorded. So you take that signal and just do a cross correlation with what you've recorded. You highlight things that have that same sound signature, those same frequencies in it, and you suppress things that don't. Things that don't tend to be things like white noise, right? That has too many frequencies. So you've improved your signal to noise ratio by looking for things that look like the signal you generate. You also get improved range resolution because of that. So you can, what it means is that if you have two targets close together, you can actually tell them apart in broadband signal processing. But in narrowband, they would look like the same target, one large target together. And you can actually see that right here. So this is an example of, on the top, single frequency sound at 18 kilohertz. And the lower one is a broadband chirp from, it's about 12 to 23 kilohertz. Same section of water. These are back-to-back pings. You can see that in the narrowband, the fish appear. These are fish. It's I call it an inverted smiley face because I think frowny face is too sad. So the inverted smiley face, you see that it's much wider in the CW pulse, and some of these are starting to get muddled together. You also see this speckling over here, which is noise. There's higher noise. But when you do an FM pulse and do matched filter processing, again, looking for that same signal, you get a thinner looking fish. You're able to discern the different fish. You're able to discern those targets as separate. And you don't have that same higher amplitude noise in the background. You have an improved view of what's in the ocean. For integrating these systems into, especially into smaller platforms. So as we start using USVs, AUVs, ROVs, all these deployed systems with a lot of electronics jammed into one place, you can end up with noise sources from all those electronics being together. And with a broadband system, you can filter out those if they're very narrowband. What I mean is that if you had 120 kilohertz sonar and you had a noise source at 120 kilohertz, kind of screwed. Now, if you have a broadband pulse that actually goes from 80 to 150 kilohertz, you can just filter out that spike at 120. You don't need to worry about it. You can filter it out in post, or you can actually filter it out in the signal you're generating too. But the most powerful part of broadband acoustics is in the ability, this is a fancy way of saying it, is reducing target ambiguity, which is where you can identify what you're looking at in the ocean just based off of how sound comes back from it across that broadband pulse. And that's what my entire career has been, basically, figuring out what stuff is with broadband acoustics. And here's an example of how powerful that tool is. So this is a figure from Kelly Benoit Bird and Chad Wallach at Monterey Bay Aquarium Research Institute. They're looking at 3 different species: krill, anchovy, and hake. The figure on the left here is what we call multispectral. So that is using 5 echo sounders, each at a distinct frequency. So there's an 18 kilohertz sonar, a 38, 70, 120, 200. Those are the frequencies, and it's just that single frequency. Comparing that with broadband, where instead we do a broad range of frequencies about those center frequencies. So instead of 18 kilohertz, we do 12 to 23. Instead of 70 kilohertz, you go from 55 to 110. You do a whole range of frequencies. And you can tell here that discerning those three species, you can kind of see, you kind of know the hake, the anchovy, and the krill, but there's a lot of overlap. It gets hard to discern those. And that has a lot to do with how many points you're fitting, basically. The way you do species identification, you take your acoustic signal, you fit it to a model. The more frequencies you have, the more points you're fitting, the more statistically rigorous your analysis is. And so when you do that, you're looking at hake, anchovy, and krill as separate groups. You can tell those apart just from the acoustic response across all those frequencies. So there's a lot of power to using broadband acoustics. So how do we do that? Again, I said this is sort of the underwater acoustics engineering side. So there are equations. I'm sorry. It's all about the wave equation, right? That's what we use. This is how we figure out what's happening in the ocean. We take this relatively simple equation. We simplify the crap out of it because I can't solve that equation myself. And we figure out what's going on in the ocean. We figure out this is what it should look like. This is what our model would say sound should look like as it scatters off of a fish, a bubble, a krill, whatever it is. We drive it from some pretty simple principles. We take the Navier-Stokes equation, which a lot of fluids folks know that one, my favorite equation. You can apply some Newton's second law, continuity, Hooke's law, a lot of these, sort of Newtonian solutions. And you get down to the wave equation. The other things to consider when doing math of waves are refraction and interference. And these are the really important parts of understanding conceptually how we solve the wave equation and how we figure out what a target is using sound. So refraction, as you probably learned from Snell's law in light, is very similar with sound. As you go from a medium here, medium 1 to medium 2, the light will bend. Now the question is, according to the sound speed, the light will bend, or sound will bend, excuse me. Which one has a higher sound speed? Medium 1 or medium 2? You can tell. Drummond?
I'm not giving you 50-50.
Which way is it bending? If you look, it's slowing down as it enters, right? These lines are getting closer together. It's actually slowing as it bends into there. So it continues to go at a faster speed on this edge of the wave. Therefore, this side is slower. Not doubting myself, but yeah, that's slower. Right? So that's how you go worry about refraction as you go from one medium to another. The other is interference. So if the blue and the green wave are passing by each other, you see they're the same amplitude, but the phases change. The red wave is the sum of those two waves. And as you go, when the peaks match, you get twice the amplitude. And when the peak and the trough match, you get 0, right? So the things you need to worry about for understanding intuitively how we solve the wave equation is really refraction and interference. I'll sort of explain what I mean by that. So if we have a thing, we have a sphere. We really like to model things as spheres, right? Because it takes three-dimensional equations, makes them symmetric, and now I only have one dimension to worry about. Makes life a lot easier. What happens when a sound wave, let's call this a sound wave in the ocean, meets this bubble, this spherical bubble? What happens to the sound? Right here at the front of this, where it hits, where the medium changes, what happens to the sound? It bounces back, right? Does all of it bounce back?
That's right in the center.
It's right in the center. Here, the wave's probably larger. I don't think all of it will bounce back. I think we're all going to get some transmission through the wave. That depends on your reflection and transmission coefficient, which you figure out from the physical properties of the medium and the target you're bouncing off of. With a bubble, most of the sound bounces back. Yeah, exactly. If we're looking at a fluid sphere, something that's made of oil, then only a little bit of the sound is going to reflect and some is going to pass through, right? What determines if sound bounces off the front or not? What are density differences? That's one component. There's one other, which we just saw with the refraction example. The sound speed. Those two combine to give you the specific acoustic impedance, which is one of those word envy things that scientists come up with for just the product of density and sound speed. That determines how much sound bounces off the front. Now, what about some of that sound that transmitted into this bubble or oil droplet? What about this end? What's going to happen? It bounces again, right? It's the same contrast. It's just a different direction. Some of that sound is going to bounce off the back as well and come back. This is where that constructive and destructive interference becomes really important. So if the path length difference, so the sound bounces here, some bounces back here, if that is equal to half a wavelength, you're going to get perfectly destructive interference. The waves are going to come back completely 180 degrees separate from each other and cancel each other out. Now, the frequency is what determines if that happens. The wavelength is determined by the frequency of sound you generate, right? So if you change the frequency, you change how the interference works. And that's why you need broadband. It's very different depending on the frequency you use. Now, it turns out there's all kinds of other cool waves. There's an evanescent wave, which is not a band from the 90s. It's a sound that moves around the edge and then decays that moves away from the edge of the sphere. There's a whispering gallery wave, which is actually a really cool wave. If you've ever been to Grand Central Station in New York, there's an area that's like sort of like a dome, or any dome really, been on the other side. You go to a corner, you can whisper in the corner, and the sound shows up on the other side as if you were talking directly to that person. That's a whispering gallery wave. It hangs out on the inside of the boundary and then comes back out. So there's actually a whole bunch of different things to consider when you figure out how the things interfere with each other, how they refract, what kind of interference you get. But in the end, it is solvable in some cases. And you can figure out what you're looking at based on the sound. So on the y-axis here is we call the volume backscattering. So this is just basically the amount of sound that scatters at a given frequency. And these here are some of the more common frequencies used in acoustics. So 120 kilohertz and 200 are pretty ubiquitous. 43 is common for some systems, 38 is common for another. It's just sort of showing you these different species. A fish. Fish have this great big resonance peak because they have this is a swim bladder fish. Swim bladder will actually resonate, which means when you hit it with sound, it vibrates and generates sound itself. So you get pretty high amplitude scatter at that frequency. With a squid, pretty big, but no resonance peak because there's no gas inside of a squid for the most part. This is turbulent microstructure, so you actually get scattering off of turbulence between two layers in the ocean. As two layers go past each other, they generate these turbulent microstructures, and you get scattering from those. So you can do physical oceanography with broadband acoustics as well. And then you sort of see all these different species that you can figure out and identify based off their frequency response. So how do we solve that? How do we deal with the wave equation? In some cases, you can actually solve it. So for a sphere, a fluid sphere especially, there is a solution. This is quite the famous paper in acoustics where Victor Anderson solved the wave equation for a fluid sphere. It's a bit cumbersome, but you can code it up. It's frustrating to code up, but you can do it and solve that situation. But oftentimes you don't have a perfect sphere, right? Actually, we seldom do. Turns out even gas bubbles and oil droplets are rarely spheres because when they rise to the water column, they squish. And you end up with an oblate spheroid, more likely. And so we do simplifying assumptions. In this case, on the left, this is something called the distorted wave Born approximation, where we slice it up into a bunch of disks that are radially symmetric. So, okay, I can solve for a flat disk here and a flat disk down here, and then I just add all those together, and I get the scattering from that oblate spheroid. In other cases, we can get even more analytical. This is a method called the boundary element method, where you actually model the boundary itself in small chunks, and then add all of those together. And that's a computationally intensive way of doing things, but it's very close to a precise solution. So you either solve the wave equation when you can, or you use some simplifying assumptions, or a big computer. And then you get to the answer of what the scattering should look like, and you come up with figures like this. The critical part before we jump into the acoustical oceanography of actually using this stuff is that you have to validate the model. Doing the math and doing the assumptions is great. But you should actually make those measurements, typically in a lab, and see if they agree with what your model says. And to do that, we usually take a dead frozen squid and put it in a tank. This is a study by Woo-Jung Lee, who's now at University of Washington, where she took a squid, hung it from this marionette-like structure, spun it around, and looked at how the sound scattered off of this squid based off of the angle of the squid relative to the sonar. So you can imagine if you've got a fish swimming at you, you're going to get some sound bouncing back. If you've got a fish swimming parallel to you, you'll probably get more. So the orientation of what you're looking at also matters. So we do this for all the different kinds of models. Well, we try to do this to validate models. It's been done for butterfish, for hake, squid. There's a whole bunch of different models that have been validated in this way. So once you get to that point, now we've validated our model. The math we've done agrees with what we can actually physically measure in the lab. Then we can go out and use it in the field. And using it in the field can be done in a whole bunch of different ways. You can put these suckers, these broadband acoustic echo sounders, on research ships is where they started. That's where they've always sort of started their life. But we put them on moorings, AUVs, ROVs, autonomous sailing platforms, bottom landers, gliders, all kinds of different things. These things are designed in a number of different packages to be used in a number of different ways to measure a bunch of different stuff. Typically, the bread and butter is biomass. You look at fish, plankton, jellies, whatever you want to figure out the biomass of, you can go and use acoustics. And I'll show an example of one of those in one of my papers. There's also non-biological targets. I talked about the turbulent layers you can measure, the turbulent structures. You can look at oceanographic layers. You can measure sediment, gas bubbles. My PhD thesis was on oil spill. You can measure oil flow rates with acoustics, all kinds of fun stuff. And you do that with a whole bunch of different kinds of transceivers, basically. So this is an echo sounder consists of a transceiver, the electronics that generate sound, and the transducer, which is what is the ceramic that actually makes the sound. So this is the computation side. There's rack-mounted versions, there's autonomous versions, things for putting on moorings, things for small boats. All kinds of these all exist. You can put them on whatever you want. And there's a whole bunch of different transducers. The reason I say this is that the sound you generate depends on your transducer. If you want to make low frequency sound, you need a big transducer. If you want to make high frequency sound, you need a smaller transducer. So that's why we talked about that multispectral acoustics, where you use a bunch of different single frequencies. You had to use different transducers to do that, to generate that sound. Okay. So now we talk all about that. We talk about the power of broadband acoustics, basically. So this is a paper about the Challenger Deep in the Mariana Trench. If you don't know, there's the Mariana Trench right off Japan. In the Challenger Deep, there are effectively 3 different spots that all claim to be the deepest part of the ocean. I think you may know Victor Vescovo recently dove down to those in his crude submersible. There's been a whole bunch of different ways of trying to measure what that deepest point is. And none of them entirely agree. These are all the different estimates of the depth of the Challenger Deep. So originally in 1952, the HMS Challenger 2 came back with a lead line and measured the depth of the Challenger Deep. Interestingly, the Challenger 1, for which the deepest spot is named after, was also lead lining, and they got to a point in the ocean where they didn't have enough line, which must have been terrifying, right? Like you're on an exploratory experiment trying to figure out the depth of the ocean, you find a part where there's just a big hole. That would freak me out. The depth you will see most commonly referenced in anything, especially popular media, National Geographic, that kind of thing, is this estimate by the RV Vityaz, a Russian vessel, in 1957, which is just over 11,000 kilometers. And it's wrong. This was done with an average water column sound speed. If you've ever taken a profile of the sound speed in the ocean, it is not constant. It's a big old curve. And so there's a huge error in this. In, which one was it? I don't think it was one of these estimates. Oh, come on, who was it? I'm blanking. Oh, Korea. Oh, yeah. Korea, or I'm sure I'm butchering that name. In 2002, went back, did a proper sound speed profile, and applied that to the timing measured by the Vityaz, and measured a depth of just over 10,900 kilometers. 10,900 meters. So it's wrong. I think we can all agree on that. And all of these are the other estimates. The single point measures are typically from ROVs, or I couldn't find any statistical analysis in the paper. Interestingly enough, the Vityaz paper that's so referenced, I had to find a Russian speaker to translate the paper to tell me how those measurements are made. It's never actually been translated in English. These point measures basically don't have any statistics about them. For this study that I'm going to talk about, we have one of the narrowest measures of the depth of the Challenger Deep and one of the deepest. And it was all done by accident. So 2 passive acoustic platforms were deployed in the Challenger Deep in 2012. One of them imploded on its way down. There was a flaw in the glass sphere and it imploded. The second one recorded that implosion. And in 2020, when it was COVID and no one could go out to sea and get any data, I needed to do something. And so the guy who had this data set sent it to me, and I messed around with it, and we made a measurement of the deepest part of the ocean. So the sphere is a glass sphere with all the computational, all the computer components inside. There were 4 hydrophones on the outside, a CTD, which this is actually critical. It made direct measurements of either the CTD and the sound velocity as the probe is being dropped in the water column. It turns out the time between which you make a sounding and when you made your sound speed water column measurement really matters. You have significant errors if you wait a year to do the CTD cast, because it's not applicable to when you made the sound. And that's in some cases people have had to do that. So this was a direct measurement of the sound speed at the moment when the sounding was made, which is part of the reason why we were able to get such a narrow estimate. The other is that it was a very broadband signal. And I can play that sound, I hope. Come on now. So there's the initial implosion. And if you listen, you can hear the echoes. That's the first echo. Another one right after it. Another echo. If you really listen closely, at 30 seconds after the initial implosion. Right there. Another echo. The average sound speed in the ocean is about 1,500 meters per second. 30 seconds is a long distance for sound to travel. Turns out that's multiple reflections in the Challenger Deep off the bottom and the surface. This is what happened. We had our receiving array. It was at a depth of about, turn on my laser pointer again, 8,259 meters. We figured out from the sinking rate of these platforms, the source was at about just over 9,000 meters. And then you have all these different paths for the sound to travel. Does anyone know how I can get rid of that thing? Okay. That works, right? Okay. That works for me.
Okay.
All right, go back here now. Okay.
Yeah, it might come back.
Okay. I hope it doesn't. Okay. So if you take these different paths and break them down into basically the path lengths you go. So a reflection off of the bottom, that's what this is, the pressure bounced off the bottom. It's Z1 plus the difference between Z and Z, so this distance, plus the sinking rate of the platform and the time between the arrivals, right? You have to account, over 30 seconds, you certainly have to account for how far your platform has sunk while you're doing this equation. This is where the broadband acoustic side comes in. At the top, the gray lines are the actual recorded signal. Now, critically, it clipped at the initial arrival. So when the first wave arrived from the implosion, it was such high amplitude that it saturated the hydrophone, which actually was a big problem because we want to use that signal that we think it looks like to do that matched filter processing, to do that cross correlation, look for the signal of the sound bouncing around. Now, luckily, we had another return, which is lower amplitude, but was isolated. So we can use that as our matched filter. We take a look at, this is the gray line showing the frequency response of that section that's highlighted in red above. So this is the frequency by the power. And then we do a little bit of filtering. So look at just certain frequencies. We look at this really unique structure, this peak and null structure, that's pretty common to an implosion in the signal. So we're going to use just this red component as a matched filter and multiply and cross-correlate it with the signal. Now, the reason that it has sort of that funny shape and frequency space is that when you make an implosion, when an implosion happens, you make a big bubble. And that bubble shrinks down to almost nothing, and then it expands. It goes back and forth and bounces like this. And it creates this radiating sound. When you do the cross-correlation, you can't use anything in the clipped section because the frequency content's all a mess there. You get these peaks. You get a peak from the bottom reflection, So that's the sound that bounced off the bottom, a peak off the surface, a peak that reflected off the bottom and then the surface, and then came to the receiving platform, which remember is about 8,000 meters deep. And then the final one bounced off the surface, the bottom, the surface again, and then was recorded by the receiving platform. Traveled a really great distance. But you see how peaky these are. So we get a really clear view of when in time the signals arrive by using matched filter processing. But what's really exciting is we only have these returns. And it's easy to explain why. Basically, this was the direct one that clipped. This one basically arrives at the same time as this one. The one that went off the surface and the bottom arrives at pretty much the same time as the bottom and the surface. So it's, it's essentially the same arrival. This one that reflected off the bottom was really, really low in amplitude. It's because the bottom of the ocean is a much poorer reflector of sound than the surface. So more sound went through, more sound got dissipated off of the double bottom bounce. The double surface return, however, arrived. And what's really nice is if you take the difference between the bottom surface arrival and the surface arrival, you're left with just 2 times the depth, the sinking speed and the time between those arrivals. So you don't need the initial arrival. You don't need T0, basically. You can just take the difference in time between those 2 things arrived, and then you know the depth. Now, it's a bit of an oversimplification, right? Sound travels in rays in the ocean. It doesn't just go straight back and forth. The platforms are separated by a known distance. So we did some modeling using the sound speed profile to do the actual ray tracing. And that actually changed our estimate by about a meter. So it wasn't that significant. If you remember, this is about 11 kilometers of depth, and they were separated by an order of less than a kilometer. The angles are quite small. But in the end, the depth of challenge deep is 10,983 plus or minus 6 meters. Percentage-wise, that's a pretty small uncertainty. All that from just doing matched filter processing, from doing broadband acoustics. So I know that's not a sonar, technically. I guess it is. I generated the sound and I recorded it, but I didn't mean to generate sound. All right. So the next way we've used broadband acoustics is to do species identification and biomass estimation of an aggregation of species at the New England shelf break front. So on the East Coast, there's a big, long, gradual shelf, right? And then it suddenly drops off the slope. And there's some really cool acoustics that happens out there. This was part of a ONR research we did at Hui, and it's a nice little humble brag. It was the cover of the Journal of the Acoustical Society of America. And we're looking at this aggregation here and the front. This is actually the frontal boundary of 2 water masses meeting. So the study area was right south of Woods Hole, near the Atlantis and Explorer trenches, or I'm sorry, canyons, right off of the shelf. Now, this is a project funded by the Navy in support of something called the Acoustic Telescope, which is a big passive array that effectively is a submarine hunter. So why they want to know about fish is that those big fish schools can actually change how your sound moves through the ocean. So if you're the Navy looking for submarines, you want to know if it's fish bouncing the sound around or a submarine, right? So they want to know what the ambient environment looks like. So we're out there looking at the ambient environment, which, as a side note, is when you want to do oceanography in an environment that might mostly be funded by defense, you come up with clever excuses like it might bounce your sound around when you're looking for a submarine, let me go look at the fish. Might be relevant for the next 4 years. At the shelf break, you have 2 water masses. You have the warm, salty water, warm, less saline water of the slope, meeting the shelf water, which is colder and more saline. Those 2 water masses meet, they create a front, all kinds of cool stuff happens. You get flow towards us, that's the flow here. The wind is typically going into the page here. You got bottom flow that moves nutrients over the shelf, all kinds of cool stuff. You can actually see it in the acoustics. So this is the broadband acoustic signal. There's your seafloor right there, dropping off at the slope. And this here is the frontal boundary. This is the 2 water masses meeting. And this is that big aggregation of scatter. So we were going out there to study the front and didn't know that big aggregation of scatterers was going to be there. So we didn't have a net. We didn't have a camera. We didn't have any other way to figure out what that stuff was, but we wanted to know. And this is where broadband acoustics becomes a really powerful tool. So what you see depends on the frequency. Sorry, these are very small, but these are 18 kilohertz, 38, 70, 120, and 200. Those are the center frequencies of the transducers. All at the same time, looking at the same water mass. And you can see that looks very different. Up in these higher frequencies, you're seeing the plankton that hangs out at the boundary. They start scattering sound. Down at low frequency, the plankton doesn't really scatter sound. It's more the boundary itself. So you're seeing scattering from the turbulence. Point is, you can identify what you're looking at based on the frequency, even just visually. When we start getting into the actual data analysis, you can really start to tell what you're looking at. So we had a bunch of transects over these aggregations. What was kind of cool is they didn't move. They kind of stayed here, even as the frontal boundary, which is where the front met the seafloor, shifted back and forth. The biology actually stayed in the same place, which I thought was kind of interesting. I thought it might follow that front. We did a little bit of image processing, separated the aggregation from the echogram and isolated just the biological targets. Looked at the broadband backscatter. So this is again that frequency by amplitude plot on the RV Armstrong that we're on that had narrowband 18 and 38 kilohertz. It is now possible to get those in broadband, but at the time when the vessel was outfitted, they were only available in narrowband. And from that backscatter, we were able to tell that what was at the front was a nursery of mackerel and squid, small mackerel and long fin squid. So how do we figure that out? We had to start with a cast of likely candidates. So we used the NOAA Northeast Fisheries Science Center trawl data, which these are random trawls in the area that kind of tell you what's likely to be present in that region. They weren't targeting those aggregations at the front. So what was in the net wasn't necessarily what we were looking at, but we know what's in the area, what's likely to be there. And so long fin squid, the most common, kind of expected that. Then butterfish, dogfish. The red species all have swim bladders. The scattering did not look like a swim bladder fish, so we eliminated those right away. And then the blue species are flounder, skate, demersal fishes. They're unlikely to be tens of kilometers up in the water column. So we can limit our analysis to looking at longfin squid, butterfish, dogfish, or mackerel. So already we've limited things down to the likely candidates. The backscatter from those species looks pretty different. So this would be the swim bladdered fish. We can eliminate that right away. It wasn't very high scattering and low frequencies. The longfin squid has this sort of same peak and null structure. It's rather simple when compared with butterfish, dogfish, and mackerel. That's much more complicated. You actually have to model the scattering off of the spinal column of the butterfish and the dogfish, as well as the body. Sorry, off the butterfish and the mackerel. And the dogfish, you have to model the liver and the body of the dogfish to get the scattering. And we did that with the distribution of sizes for those species at the shelf break. We got the dashed line, which does not fit very well to our measured acoustic response. We have to remember those trawls are not targeting the area we're interested in. They're just random trawls of the region. So if we do a little bit more analysis, we modify our size distribution, we can find scattering that fits with what we measured. So what we did was took the trawl data size distribution of those species, iteratively changed the distribution and the ratio of species until we had a model that converged globally to the scattering we had recorded. It's critical that it converges globally, right? If you have many minima, many places where the sound fits, you have an underdefined system. It could be all kinds of different things. But the fact was the sound actually converged to a single solution, which gives you that confidence that at least you're close to what the actual distribution is. Why wasn't it butterfish and squid? To fit the scattering would have had to be the largest butterfish anyone has ever seen in their entire life. There's never been a butterfish caught this large in the history of the NOAA Northeast Fisheries Science Center. So it's probably not a secret land of the giant butterfish. With dogfish, it started to converge to just squid. We had that ratio term, which was the ratio of dogfish to squid. And as you lowered that ratio, so it was becoming more and more just squid, it got closer and closer. The root mean square error got smaller and smaller. So that means that it's unlikely dogfish are significantly contributing to the scattering, right? But when we use mackerel and squid, it converged to a decent ratio, mostly squid, but a lot of mackerel too, which already tells you that it fits better than dogfish and squid, right? You think about it. If squid fit better than a squid with a dogfish, and the mixture of mackerel and squid fit better than just squid, this fits better than the dogfish and squid solution. The mackerel are quite small, not impossibly small. They start quite small, right? This is sort of a nursery of mackerel and squid at the shelf break. And then that all converged to a single global minimum solution. So what we have is mackerel and squid at the front. What's interesting is, you can kind of ignore this one. This is showing that there's no pattern based on the cruise tracks we went on. This is looking at the biomass. So this is a biomass estimated by using the size distribution from the trawl net compared with the size distribution from the matching method we use, where we iteratively change the size of the two species. And this is unity. So you see that we're actually getting a substantially higher biomass in the model fitting version. So using the size distribution from the trawl net would likely underestimate the biomass at the shelf break front. It's a whole lot of little ones instead of a few big ones, basically, when it comes down to it. So a couple more minutes. What about those frontal boundaries we saw? Can we do anything cool with that? And so this is from a paper that we just published a couple months ago, actually. Looking at those frontal boundaries to use the broadband echo sounder essentially as a CTD. So when you do the location here of these vertical yellow lines is the location of CTD casts we did on later cruises. This is from a different data set than the mackerel and squid paper, but at the same location. So we did a bunch of CTD casts through these regions. And you can see the frontal boundary here in the broadband echo sounder, or in the 18 kilohertz echo sounder. And typically what you would do is you take those CTD casts and you do an interpolation between them, right? You do two-dimensional interpolation to figure out what's happening between those casts. This is typically how we do CTD profiling. You don't actually know what's going on in between those casts, you're guessing. An educated guess, typically, but you're guessing. Can we fill in those gaps with the acoustics to actually show in high resolution what's happening between the CTDs? And the answer is yes. So we take this profile, trace out those boundaries, and then use those as a guide. Now, in this case, we used a single CTD. So we did is we took this CTD cast that went through all the water masses, used these lines to project that north and south of that location, and create this high resolution map of the sound speed in this case. You can do temperature, you can do salinity, you can do any property that maps to those water masses that you're able to identify acoustically. So this is the profile done with the interpolation between casts, and then this is using the EK, the echo sounder, and a single CTD cast. And what's critical is that we can then use these other CTD casts as ground truthing and show how well does this agree, this CTD cast, this direct profile, agree with our estimate of the sound speed in this location. It agrees really well, but I don't see that cast on my slide. So that's a publication. I can pull it up if anyone's still interested in looking at that. I guess I don't have the CTD casts in this slide deck, but those are available. They're in the paper. I'm happy to show you. They agree really well, basically. That's how we got the dang thing published. The CTD casts agree really well with our inferred profile of the sound speed at the front. What's really cool is there's a bunch of internal waves, a lot of really interesting stuff that you can see in this profile that's really important to things like long-range propagation modeling people use for submarine hunting or for doing long-range measurements of the temperature to look for changes in the ambient temperature of the ocean. Operationally, this can actually be a really powerful tool. So we were out there off the shelf, and a huge storm was coming in. We had to get back to Martha's Vineyard and hide from the storm. We only had time to do one CTD cast, but what we saw in the echo sounder was crazy. Usually the front is this wedge that comes down and it makes a pretty simple profile, but we have these profile lines coming all the way to the surface, doing all this crazy stuff. What turns out is that when we got back, we looked at the sea surface temperature data and saw that there was a cyclonic eddy coming through the region. And that was pulling up welling to the surface, which is actually pulling slope water all the way up to the surface. Now, anecdotally, a lot of people have seen slope species in small pockets at the shelf break without a great explanation of why. And it might be that we're just missing it when we do these CTD profiles. You need to do a lot of CTD profiles to resolve this. But if you use the single cast and those lines drawn from the echo sounder, you get to see that whole profile and see what the sound speed looks like, or the density, or whatever property that maps to those water masses. So there's a ton of different stuff you can do with broadband acoustics. This is just not an all-inclusive list, but this is a list of some of the things it's been used for species identification, which we just talked about, and biomass estimates, sediment plume detection and mapping. That's really critical for dredging operations. Get a view of the entire dredging plume, see where that plume is going. Do you have too high of a density in the water column? Is it going to end up in a marine protected area? How do you adjust your dredging operations to protect the environment? Environmental monitoring, especially for like offshore wind stuff. Oil spill quantification, which I mentioned, was my thesis. Wake and turbulent detection and identification. It's kind of a cool one. If you've seen a lot of like sci-fi movies where they look at the thruster from the spaceship and they like identify what kind of spaceship it is based off of the thruster, right? You know? Seems like a fake thing. But you can actually do a little bit of that with broadband acoustics. You can identify things in the wake to get an idea of what kind of system is moving through the ocean. Whether it's an AUV, you can get vessel wakes, you can start basically putting a barcode on things moving through the ocean. And I'm sure it's more advanced than I know about because I don't have a clearance. There's natural seep measurements. So figuring out what the ambient flow of oil is into the Gulf of Mexico is really important. If you want to know how bad an oil spill is going to impact the community in the Gulf of Mexico, how much oil is normally there? How much can they in that environment respond to? And what is your spill relative to the ambient background level? For physical oceanography stuff, we talked about fronts and boundaries there, harmful algal bloom monitoring, seafloor carbon sequestration monitoring. So there's a lot of talk about taking CO2 off of various areas and pumping it back into the seafloor, right? Someone's got to go make sure it stays there, right? You need some way to see if that's actually seeping back up into the ocean and back up to the surface. And so there are ways to do that using broadband acoustics. You can look for bubbles, you can measure the bubble size, and you can figure out the flow rate. You figure out, is it a tiny little trickle that we don't need to worry about, or do you have a heck of a lot of CO2 coming out and you're no longer sequestering it? A lot of work being done on mesopelagic communities, because it's really hard to get a person down there, so you got to use acoustics. Hydrothermal vent work, aquaculture monitoring, fluid intake monitoring. That's kind of a cool new one where a lot of power plants that use ocean, river, whatever kind of water to cool the power plant, they'll have issues with their filters clogging at certain times of the year. So using acoustics, you can go, oh, hey, look, it's spawning season for hake. Probably not the time to take in from that pipe, take it in from the other one, or else you're going to get your filter clogged really quickly. Makes your pumps work more efficiently, makes your cooling system work more efficiently. And it just sort of tells you where to use those, which ones to use and when. There's always more. Always going to be more applications of broadband acoustics. People in this room can probably think of them right now. And that's it. I was told I had to have a whale on my slides. So these are my favorite whales, the right whales. There aren't that many of them left, especially these North Atlantic ones. And acoustics can be used to find their food source. There's a lot of people going out and figuring out the copepods, Calanus and Marchicus, that the North Atlantic right whales live off of. A lot of work finding those, figuring out where they are, and trying to use that as a predictive measure to change shipping routes. To say, this is where the whales are going to be in feeding. You need to come into Boston in a different direction now. If you can tell that to a boat in the middle of the ocean, slightly turning your tiller in the middle of the ocean, it's a heck of a lot cheaper than coming to shore, suddenly having to turn and drive all the way around. Sort of a win-win to be more predictive.
Forgot the rarest one, the North Pacific right whale.
They couldn't get a picture of them. They're so rare.
That's right.
I think we have time. I'm happy to take questions.
Thank you so very much. That was amazing. I learned a lot. We're going to jump back and forth between folks online and folks in the room. So, Roseanne, do we have any questions yet? All right, for folks online, if you have any questions, put them in that chat box and we'll get them asked in the room. How about questions in the room? I don't have to run it downstairs, so go ahead. Ask your questions. I was going to say, you stunned us all.
Can you talk a little bit more about what you're describing about the target strengths of different anatomy pieces of fish, like livers of sharks and backbones of hake or whatever it was?
Yeah. So that came from some of those experiments where they would take a captive fish, usually a dead fish, and ping on it and see what the backscatter looked like. You're like, okay, well, we're actually seeing how does this work? How does this make sense? So anytime you have a change in density and sound speed, the specific acoustic impedance, high enough, you'll get scattering. And so it turns out the vertebral column of a lot of non-gas bladder bearing fishes is enough of a contrast that you get separate scattering from that column. So there's sound that scatters off the fish, and then some of it goes into the body and scatters off of the vertebral column. It scatters off any of the bones in the fish, right? But those are the thickest, largest that contribute the most to scattering. And so the same deal with a shark. The cartilage itself doesn't really scatter very much sound relative to the body, but the lipid liver does. And so sort of you just got to figure out what those physical properties are that determine what structures within the body will scatter the most sound. Same with the swim bladder, right?
It's the noisiest part of the body.
Yeah, where there's this biggest contrast between the fish flesh itself and the vertebral column. It turns out with gas bladder bearing fish, oftentimes you can just model the gas bladder. That's such a contrast compared to everything else. You can kind of get away without doing the body. As the instruments advance and we get more and more precise with broadband acoustics, people are now reintroducing the body to that equation. So you're scattering both the gas bladder and the body. But anywhere you have enough contrast, you have to sort of account for that kind of scattering.
It's not harder to
Hang on.
account for it. It's not harder to account for it in broadband than it is in single band.
No.
Okay.
You just have to do it at each frequency because that wavelength difference is what matters, right? So
Okay.
whether you have destructive or constructive interference depends on the wavelength. So it depends on the frequency. So yeah, the hardest part is you got to get the x-rays of the fish often. And then try and translate that to different size fishes. And there's a lot of uncertainty in trying to do that.
All right, we got a question online.
How accurately can backscatter differentiate various gelatinous organisms? They look relatively close to each other on the frequency diagram.
They do. And gelatinous organisms are really bad at scattering sound. They acoustically look like water. It's kind of the design. Some species have gas inclusions. So in the example I was showing here, the siphonophore, the scattering looks a whole heck of a lot like this gas bladder bearing fish, right? It's because a lot of siphonophores actually have a small gas inclusion in them, and that's what you're scattering off of, more so than the body of the gelatinous organism. There's a recent paper by Rachel Kahn, who was at Hui, where she did direct measurements of scattering from jellies. They're not very high amplitude, but she looked at being able to differentiate the size and species. And I have to admit, I haven't looked too deep in that paper yet, but if you want to find out more, it's Rachel Kahn, K-A-H-N, from Hui. It was published last year, I think.
Great, thank you. Questions in the room?
Thanks, Scott. I was really curious about that, the result you showed where you were comparing biomass from trawl survey versus the acoustic sort of modeled estimates of biomass. And I'm curious if you could say a little bit more about what gave you the confidence to say that the trawl-based biomass estimate was an underestimate as opposed to the acoustic-based estimate being an overestimate.
Yeah. So basically what we did was, it's a good question. Let me look here. So we took the model backscatter for the size distributions from the trawl. And then in order to get it to the proper amplitude of what we measured, we added X number of fish. And if we know the size distribution and the number of them, we know the biomass. So it's based entirely off the acoustics. So whatever this number of that size distribution gave us the smallest root mean square error in that situation. And that was what we decided was the biomass estimate. And with the iteratively changed size distribution, same deal, where we had the minimum root mean square error, that's where we say this is the biomass. And every time, consistently, the iteratively changed size distribution that fit the acoustic data better gave us a higher estimate of biomass. It has to do with how the acoustic scattering scales with size compared to the volume scales with size.
Roseanne, any more questions online?
No.
Other questions in the room?
Thanks, Scott. Really interesting stuff. I would imagine that it's similar to, I mean, with this particular thing, you had two very different sort of scattering or sound signals that came back, right? But you know, I work a lot with coastal pelagic species, so it's Pacific mackerel and sardine and anchovy and herring, and they're not all that different in size and scattering. I would imagine. I'm not an acoustician by any means. So, can you speak to that and how that sort of differentiation is done? I know they do, trawls as well, but there's a long time between when they run the transect and get the, where the acoustic are, and then they come back and do the trawls.
Yeah,
a number
absolutely.
of hours later.
So the truth is that broadband acoustics can't quite give you species level identification. In this case, we had limited to likely candidates, and they were different species, and their scattering was very different. It also can't give you, we only did groups of species, right? We only did butterfish and squid, dogfish and squid, mackerel and squid. But it turns out as soon as we introduced a third species, our model no longer converged to a global minimum. It was underdefined. So there are limitations. I think that one of the ways to overcome that limitation is actually eDNA, believe it or not. If you can combine eDNA with broadband acoustics, that gives you an idea of the likely species present. I know there's a ton of questions about how eDNA works, how long-lived the DNA is in the water. There's tons more to learn, right? But if you have that targeting your likely species distribution, instead of the trawl nets, in this case done from all over the coast, we focused on a specific area, but they're not quite representing exactly that aggregation. If you were to have your eDNA sampler and your acoustics done concurrently, then you have a much more precise likely group of candidates. I think that might be a way to get even finer resolution. Maybe not species. I don't know where in the chain you'll end up. Genus would be great, but we'll sort of see at what point you start differentiating things acoustically. So the truth is, species that look very similar acoustically, they look similar.
All right, I know we're right at time, but we have one more question, then we'll wrap up.
It seems like all of the questions have kind of circled around ground truthing. Whenever you do remote sensing, you really need to do ground truthing. In the 70s, we used a 200 kilohertz echo sounder to study euphalsids in the Arctic for the deep scattering layer. But we also followed it up with frantic net sampling to make sure we actually were measuring euphalsids. Have you considered possibly using submarine cameras or something to identify do some ground truthing, back up your models with a little bit of ground truth?
That's my favorite part about this paper is there is no ground truthing, so no one can talk about it. But you're absolutely right. There are towed camera systems like the ODFW guys have that would be a great thing to use for some of this stuff. The truth is with nets, as we've seen in a number of studies, is that they can dramatically underestimate biomass and miss a lot of species. There's both catchability and selectability from nets that can really bias what you think you're seeing. That's why I lean more towards eDNA, which is a less invasive thing. So I think things we're less likely to dodge the eDNA sampler. But the question starts becoming, which one is the ground truth, right? Because the net itself has biases that we need to account for. And so acoustics is not invasive, right? You're less likely to have species avoiding an acoustic wave from a surface vessel at a given depth, like the mesopelagic there or something like that. But it's not foolproof, right? Because we also have to ground truth those acoustics somehow. And that's, we have this giant tow sled that at Hui that has multiple cameras and a whole bunch of acoustics on it and all kinds of fun stuff. But unfortunately, according to the cameras, there are no fish in the ocean. So there are biases to what you're trying to use to ground truth your system, and I don't know which one's the right one.
All right, I think we're going to wrap up with, we just don't know which one's the right one. Everybody, thank you so much for being here. One more round. All right, everybody enjoy your holiday, and we'll see you again when Jordan speaks at the beginning of December. Thanks, everyone. That's right. I love it. Oh, yeah.
Oh, I know. Right next to the Great. It's like you said, like broadband, they know what it's like.
So we have sideways looking at.
I was glad you were there.
There's one on.
It would have been hard to be the only person. Like, that's why I didn't speak up.
And so that is capable of doing exactly what you're saying, looking up the side and trying to.
I just, you were 5 minutes out, you and I said, okay, 15 minutes, we're gonna call it.